Methods for plankton research. So what are plankton? Plankton are marine drifters organisms, carried along by tides and currents. The word plankton comes from the Greek for drifter or wanderer. An organism is considered plankton if it is carried by tides and currents, and cannot swim well enough to move against these forces. Macroscopic or microscopic more than 1 inch in length. Basic categories divide plankton into two groups: phytoplankton, plants; zooplankton, animals. Phytoplankton are microscopic plants, but they play a huge role in the marine food web, like plants on land. Phytoplankton performs photosynthesis to convert the sun's rays into energy to support them, and they take in carbon dioxide and produce oxygen. Because they need the sun's energy, phytoplankton are found near the water's surface. Zooplankton include microscopic animals, krill, sea snails, pelagic worms, and etc. The young of larger invertebrates and fish, and weak swimmers like jellyfish. Most zooplankton eat phytoplankton, and most are in turn eaten by larger animals or by each other.
Sampling equipment for collecting planktons. Plankton nets, sampling bottles, and Van Dorn samplers are used to collect plankton, but they differ in their application and purpose. Plankton nets are used both vertical and horizontal sampling. The sampling bottles and Van Dorn samplers are for desired or different depth, often in stratified waters. It disappears, it indicates the density of phytoplankton.
Phytoplankton sampling method, Sachi depth measurement, maximum sampling depth or MSD calculation. We have one step-by-step process. Step 1. Lower the Sachi depth in the water column from the shadowy side of the boat, vertically until it disappears completely, and at that point, note the depth by looking at the reading on the rope at water surface. Step 2. Lower the line an additional 10 to 20 centimeter depth and then raise the desk until it reappears, and at that point, note the depth by looking the reading in the line at the water surface. Step 3. The actual Sachi depth is located at the midpoint between the point of disappearance and point of reappearance. Step 4. Report the value to the nearest centimeter and write it down in the field datasheet. Step 5. Multiply the Sachi depth with 2.5 to calculate the hypothetic zone of the lake. This is the maximum sampling depth or MSD. The last step, report the maximum sampling depth MSD to the nearest meter. Collect a planktonette sample or qualitative sample. We have 9 steps to do this planktonette sample. Step 1. Rinse the planktonette and the code end with lake water. Step 2. Set the top of the code end to the closed position and screw the code end to the net. Step 3. Tow the plankton net from MSD to the surface vertically. Step 4. Set the code end top to open position to empty the code end in the sample bottle and rinse the code end to collect all leftovers. Step 5. Pull the anchor up and start the boat engine to its minimum speed or very slow. Step 6. Collect surface sample with phytoplankton net by performing a horizontal towing of the plankton net. To do this, hold the net by the side of the boat while the boat is moving slowly for approximately 1 to 5 minutes. Step 7. Set the code end top to open position to collect the water in a plastic sampling bottle. Step 8. Label the sample bottles with the station code and sampling point number. The lake name mark PNS for plankton net sample. Date and time of collection and the collector's name initial letters. The last step, step 9. Place the sample bottle in the cooler or wrap them with aluminum foil to protect them from light and place them in a as cold as possible place. Zooplankton collection methods. We have vertical net tow. We have 8 steps to collect a vertical net tow. Step 1. Select net with approximate mesh size. Step 2. Attach net to a rope with marked depth intervals. Step 3. Lower the net vertically to the target depth. Step 4. Wait a few seconds to allow it to stabilize. Step 5. Pull the net upward slowly and steadily at 0.5 to 1 meter per second. Step 6. Retrieve the net and rinse it from the outside with clean water to concentrate. Step 7. Empty code end and contents into a sample container. Step 8. Reserve the sample net immediately. The horizontal net tow. surface or subsurface collection during the boat movement. We have 7 steps to do this. Step 1. Attach net with a flow meter to a towing line. Step 2. Deploy the net overboard and let it trail behind the boat. Step 3. Tow at slow speed 1 to 2 knots for a specific time or distance. Step 4. Retrieve net carefully to avoid sample loose. Step 5. Rinse the net from the outside into crude end. Step 6. Transfer sample from crude end to container. And last step, step 7, preserve sample properly. And we have also oblique net tow. Sample throw a range of depth in one tow. We have 6 steps on this. Step 1, set up net and flow meter. Step 2, lower the net to maximum sampling depth at an angle. Step 3, tow net diagonally upwards toward the surface at constant speed. Step 4, ensure full depth range is sampled during retrieval. Step 5, rinse and collect sample from crude end. And the last step is step 6, preserve immediately after collection.
Is counting method. In counting method for plankton data analysis, it uses binocular compound microscope. Binocular compound microscope is used for counting plankton with different eyepieces, such as 10x, 20x, 40x, and 100x. For phytoplankton, it uses 40x and 100x objectives, and for zooplankton, 4x and 10x objective. Phytoplankton uses lucky drop count method, and for zooplankton it uses the Sidgwick rafter counting cell that has a volume of 1 ml for observation. The cover slip, slide, and the Sidgwick rafter cell is used for counting the plankton's volume. For counting units for phytoplankton, the number of organisms per 100 ml of water sample is calculated using the formula below. The number of individuals per 100 ml is equal to C stands for number of organisms counted, and it is multiplied by 80, the area of cover slip in millimeters squared, and multiplied by 10. And it is divided by the AS, the area of one strip or in millimeter squared, and multiplied by S stands for number of strip counted, and multiplied by V stands for volume of sample under cover slip in milliliter. For zooplankton, it uses the formula below. Number of individuals per cubic meter is equal to C stands for number of organisms counted multiplied by V single prime stands for volume of the concentrated sample, and it is divided by V double prime volume counted in ml and multiplied by V triple prime the volume of grabbed sample in cubic meter. To obtain organisms per liter, divide by 1000. The V single prime, the volume of concentrated sample, is the total volume of the water sample after you remove the most of the extra water to make the organisms more concentrated. It is less in the water for easier to count the plankton. And the volume counted, it is the amount of concentrated sample you use for counting under the microscope. Like for example, you have a 50 ml concentrated sample, then you'll take a 1 ml to count under the microscope, then your volume is equal to 1 ml. For volume, for the grab sample in cubic meter, it is the total amount of water you collected for plankton study. For example, you collected 100 liters, then you'll convert it to cubic meter. So 0.1 cubic meter is your volume of the grab sample. The analysis, there are types of biodiversity indices. First is the species richness, species evenness, the Simpson index, and the Shannon-Weiner index. The species richness is the total number of species found in an environment or a sample. The species evenness is a measure of how similar the abundance of different species are in a community. Evenness range from 0 to 1. When evenness is close to 0, it indicates that most of the individual belong to one species or categories. And when evenness is close to 1, it indicates that each species consists of the same number of individuals. In Simpson Diversity Index, it ranges 0 to 1. The greater the value, the greater the sample diversity. This represents the probability that individuals randomly selected from a sample will belong to different species. Using the formula of D is equal to small letter n and divided by the big letter N squared. Or it could be D, the sum of small letter n minus 1 divided by big letter n minus 1. The small n represents the total number of organisms of a particular species, and the big N represents the total number of the organisms of all species. The value of D ranges between 0 to 1. Simpson's index scale level of biodiversity indicates that 1 represents no diversity, and if it is close to 0, it has a diversity in that particular area. Next is the Shannon-Weiner Diversity Index. It is calculated using the formula of H stands for value of Shannon-Weiner Diversity Index, and P stands for proportion of the individuals that belongs to a specific species within a total number of individuals in a given sample. And the log is a natural logarithm of P. It measures the biodiversity of a system by considering the species richness and evenness. And S stands for the number of species in a community, a total number of species within a sample. Typical values are generally between 1.5 and 3.5 in the most ecological studies, and the index is rarely greater than 4. The Shannon-Weiner index increases as both the richness and the evenness of the community increases. The Shannon-Weiner Diversity Index is better than the Simpson Index. It is because it provides more comprehensive measure of biodiversity by considering the both of species richness and species evenness.